Drug candidate for reversing mucosal barrier damage by HIV-A vaginal microbicide that could prevent sexual transmission of HIV-1 in women has tremendous potential for saving lives and helping staunch an epidemic. But the delicate milieu of microbes that naturally colonize the vagina and maintain a healthy mucosal barrier have made the search for such a microbicide challenging, thwarting clinical trials of the most promising candidates. Investigators at Brigham and Women's Hospital led by Raina Fikorova, MD, PhD, in collaboration with an international team, tested a laboratory-made version of a naturally occurring protein, recombinant fragment of human surfactant protein D or RFHSPD, on bioengineered vaginal tissues, immune cells and microbes to determine if the drug candidate could help prevent HIV transmission safely. In a paper published in Frontiers in Immunology, the team reports on the technique's success in preclinical testing, establishing it as a promising anti-HIV-1 vaginal microbicide. To find a new drug that can most effectively help the immune system without causing adverse effects is a formidable task that requires both fundamental understanding of the mucosal ecosystem and biotechnological know-how, said Raina Fikorova, MD, PhD, director of the Brigham's Laboratory of Genital Tract Biology. Our team was able to combine expertise in both of these areas to carry out this work and build a strong case for clinical trials of RFHSPD as a topical anti-HIV microbicide. The team designed a model of sexual transmission of HIV through bioengineered, multi-layered vaginal tissue toward the underlying white blood cells. Two major events generally lead to HIV infiltration and transmission: inflammation and the breach of the mucosal barrier. The research team reports, for the first time, a genetic signature that is tied to this breach. The activity of genes involved in inflammation and the tightness of the barrier were altered in the presence of the virus, helping promote infection. Fikorova and colleagues found that the drug candidate RFHSPD not only prevented HIV transmission, but also reversed the genetic signature of vaginal tissue in the virus presence. The molecule helped maintain vaginal tissue integrity and suppressed a pro-inflammatory response, preventing the virus from getting a foothold. In addition, multiple preclinical tests showed that the molecule could safely be applied without altering naturally occurring bacteria such as lactobacilli or disrupting vaginal mucosa. Our study offers a highly reproducible, cost-effective model to study the effectiveness of candidate microbicides. Our results provide a strong argument for further evaluation of RFHSPD as a potential topical anti-HIV microbicide. Viral peptides critical to natural HIV control. Investigators at the Ragon Institute of MGH, MIT and Harvard have used a novel approach to identify specific amino acids in the protein structure of HIV that appear critical to the ability of the virus to function and replicate. They also have found that the immune systems of individuals naturally able to control HIV infection target these amino acids with pathogen-killing CD8 T cells, an ability seen even in controllers who do not carry versions of the HLA-B protein previously associated with HIV control. The report appears in the May 3 issue of Science and could guide the development of broadly protective vaccines to prevent and suppress HIV infection. Our findings provide a putative mechanism for a major component of successful immune control of HIV and help decouple HIV control from specific versions of HLA genes, which makes our finding broadly applicable to the global population, says Gaurav Gaiha, MD, DPHIL, of the Ragon Institute and the Massachusetts General Hospital MGH Gastrointestinal Unit, co-lead author of the science paper. We believe these findings could have major implications for the development of T-cell-based vaccines for HIV and for other pathogens as well. In the 25 years it has been known that a few individuals infected with HIV are naturally able to suppress the virus with their immune systems. A highly functional CD8 T-cell response has been suggested as one probable mechanism. A 2010 Ragon Institute study found that specific variants of the HLA-B protein, which carries viral peptides to the surface of infected cells and presents them to the immune system, marking the cell for destruction by CD8 T cells, were common in HIV controllers but not in individuals with progressive HIV infection. 5 amino acids in the most common control-associated variant, HLA-B57, were found in the lining of the binding pocket, the portion of the protein that grabs and displays viral peptides. But subsequent research identified a few controllers who do not carry HLA-B57, indicating that other mechanisms could be involved. For the current study, the Ragan team used structure-based network analysis, a novel approach to examine the complex, structural bonds within and among viral proteins, to identify residues, specific amino acids within a peptide, crucial to viral function. Co-led author Elizabeth Rawson, MD, PhD, of the Broad Institute and Massachusetts Eye and Ear explains, all proteins fold into their ultimate three-dimensional structure through a series of bonds between amino acids, so we can mathematically represent proteins as a network of connections between amino acids. This allows us to use an approach called network theory, a well-defined area of mathematics, to ask a large variety of questions about the structure of the network as a whole, as well as its individual parts. Applying network analysis to information obtained from the Protein Data Bank on the structure of 12 of the 15 proteins that make up HIV, the investigators were able to calculate network scores for each amino acid, reflecting the number and relative importance of bonds with other amino acids in the protein. The more highly networked an amino acid was, the less likely it was to be mutated in thousands of viral sequences taken from patients, implying that highly networked residues were important to viral function. To test that implication, the investigators engineered mutations into amino acids with high and low network scores and found that mutating highly networked residues substantially impaired the ability of HIV to infect cells and replicate. In contrast, mutating amino acids with low network scores had little or no impact on viral infectivity. The GAG-P24 protein, which is important for formation of the virus's protein shell, had the highest frequency of networked amino acids, which supports previous studies finding that it was more sensitive than other HIV proteins to the effects of mutation and that T-cell responses targeting GAG-P24 were associated with lower viral levels. The team also produced network scores for protective and risk-associated HLA variants and found that protective forms like HLA-B57 commonly presented viral peptides that were highly networked, while risk-increasing versions were more likely to present poorly networked peptides. This finding suggests that HLA-B57's greater likelihood of presenting highly networked viral peptides to the immune system could induce a CD8 T-cell response targeting sequences more critical to viral function. To investigate the relationship between these findings and natural HIV control, the team compared the CD8 T-cell responses of a group of controllers with those of individuals with intermediate viral loads and those in whom infection was progressive, as indicated by higher viral loads. Not only was the proliferation of CD8 T-cells highest in controllers, their T-cell response also was directed against highly networked amino acids, which were weakly targeted by non-controllers. CD8 T-cell targeting of highly networked peptides was also seen in controllers who do not have protective forms of HLA, indicating that the ability to mount a T-cell defense against those viral peptides may be more important than an individual's inherited HLA variants. This study implies that HIV controllers are able to keep the virus in check with highly functional CD8 T-cell responses directed against critical peptides, which cannot be mutated without costly effects on the virus's ability to replicate, allowing persistent killing of infected cells by CD8 T-cells, says Gaiha, an instructor in medicine at Harvard Medical School, HMS. We've already begun working on development of a vaccine based on these findings, incorporating highly networked peptides presented by versions of HLA that are common throughout the global population. Ragan Institute Director Bruce Walker, MD, says, in 1994 we first encountered a patient who was able to naturally control HIV with his own immune system, and now we finally understand how he and others like him are able to do it. Walker is the Philip T. and Susan M. Ragan Professor of Medicine at Harvard Medical School, a Howard Hughes Medical Institute investigator and senior author of the science paper. Developing a vaccine against Nipah virus. Nipah virus is a type of RNA virus transmitted from animals to humans. The infection causes severe respiratory illness and symptoms including cough, headache and fever, which can progress into encephalitis, seizure and coma. Currently, there is no approved vaccine against Nipah virus available on the market. In recent years, outbreaks, from bat to human or pig to human transmission, have been reported in Malaysia, Singapore, Bangladesh and India. The World Health Organization (WHO) therefore lists Nipah virus as a priority pathogen needing urgent action. In order to reduce the risk of Nipah virus becoming a global danger, it's essential to develop a safe and effective vaccine against the virus for humans and animals. Matthias J. Schnell, Ph.D., chair of the Department of Microbiology and Immunology, and his team from Jefferson Vaccine Center at Jefferson, Philadelphia University plus Thomas Jefferson University, developed a novel recombinant vaccine called NIPRAB that shows robust immunization against Nipah virus in animal models. They published their findings in NPJ Vaccines on April 15. Dr. Schnell, together with the first author and graduate student Rohan Keshwara, took advantage of a modified rabies virus vector, and incorporated a gene from Nipah virus, creating a viral particle that displays components of both viruses on its surface. The rabies vector is a well-established vaccine strain with little capacity to cause diseases in the nervous system. Because the immune system interacts with both viral components, it develops a reaction that is specific and can defend against both viruses. The researchers showed that the live vaccine was safe in mice, which had steady weight gain and no sign of neurological diseases. Dr. Schnell and colleagues demonstrated that one dose of vaccine elicited a strong antibodies response against Nipah virus and rabies virus. Those antibodies also react to a virus from the same family as Nipah, the Hendra virus, which causes similar symptoms. In addition to the live version of the vaccine, which would be ideal for use in animals, the researchers also developed a chemically killed version of the vaccine so that the viral replication was completely abolished. They found the inactivated vaccine induced as strong immunity as the live vaccine did, and would therefore be an ideal vaccine for immunocompromised individuals, such as HIV patients, pregnant women and children. We have a vaccine that is safe and effective against Nipah, Hendra, and rabies virus in mice, says Dr. Schnell. Future work will focus on testing the vaccine on different species and establishing the right dose of injection. We also use the same vaccine platform to develop vaccines against several other emerging viruses, including a vaccine against Ebola virus, which is close to entering into its first clinical trial. Opioid epidemic is increasing rates of some infectious diseases. The United States faces a converging public health crisis as the nation's opioid epidemic fuels growing rates of certain infectious diseases, including HIV, AIDS, hepatitis, heart infections, and skin and soft tissue infections. Infectious disease and substance use disorder professionals must work together to stem the mounting public health threat, according to a new commentary in the Journal of Infectious Diseases. The article was co-authored by officials from the National Institute of Allergy and Infectious Diseases (NIAID), part of the National Institutes of Health, and the Institute of Human Virology at the University of Maryland School of Medicine in Baltimore. Since 1999, nearly 400,000 people in the United States have fatally overdosed on opioid-containing drugs, with 47,600 deaths in 2017 alone. Many people with opioid use disorder (OUD), who initially were prescribed oral drugs to treat pain, now inject prescribed or illegal opioids. High-risk injection practices such as needle sharing are causing a surge in infectious diseases. Additionally, risky sexual behaviors associated with injection drug use have contributed to the spread of sexually transmitted infections. Infectious disease health professionals can play an important role in addressing the problem by not only treating a patient's injection drug use-associated infection but also connecting the patient for treatment of their underlying OUD, the authors write. For example, coupling opioid agonist therapy, such as methadone, with treatment for HIV or hepatitis C, can prevent further transmission of those viruses and reduce opioid use. Comprehensive treatment will result in improved outcomes for both the infectious disease and the underlying OUD, according to the authors. Conversely, substance use disorder health providers should screen their patients for unrecognized infectious diseases and consult with their infectious disease colleagues regarding a comprehensive treatment plan. Substance use disorder professionals also should be aware of and direct patients to needle and syringe programs, which can decrease injection risks and provide an opportunity to provide other services as well. New federal resources made available to address the growing opioid epidemic can assist health professionals in improving and implementing coordinated, evidence-based strategies to prevent and treat OOD and opioid-associated infections. These efforts will be key to stemming and ultimately ending the intertwined problem of OOD and infectious disease, according to the commentary.